Thank you and good afternoon. So I have an impossibly long title. Thanks, Pat and Kim. And so I'm going to try to do my best of giving you an overview of Duchenne, mutations, and how we might think about genetic modifiers and even find genetic modifiers for Duchenne muscular dystrophy. So the human genome is something I've been working on and studying, and the world tried to tackle decoding the first human genome starting in the late 1980s. And over the course of the next 10 years and about $3 billion, the first sequence of the human genome was decoded. And it revealed that there's about 24,000 genes and many varieties of these genes that make up each individual human. In 1986, actually, prior to the decoding of the whole human genome, the dystrophin gene was identified on the X chromosome and has led to a lot of insight about what Duchenne muscular dystrophy is. So from its first cloning in 1986, it gave us insights to what was the molecular cause, what's the root cause, and we're all quite used to this now. We all get genetic diagnosis. It wildly improved how diagnosis was performed and permitted specific genetic counseling. It also provided insights for why do some kids have a relatively more severe disease, Duchenne, and others a relatively less severe disease, Becker muscular dystrophy, and indeed some with dystrophin mutations are living and actually quite healthy into their 60s. So there's quite a spectrum of disease that can come from the mutations in this gene. And that, of course, has led to some insights about how would you go about ever doing gene therapy. Exon skipping as a strategy. Utrophin upregulation is also another genetic finding. So there's a host of such things that are really born from understanding the genetics better. Duchenne has been long recognized to be an X-linked recessive disorder. It's often inherited from the mother. The mother's a carrier, and the male child, inherits one defective allele of dystrophin. But many families, and actually I think it's certainly well above 30% of the families, the one looked out like the one down below, where the mother is not known to be affected, not known to be a carrier. The father's not affected, and nobody in their family has ever had this disease, and yet they have a boy who shockingly has Duchenne muscular dystrophy. So this is actually quite a common presentation, and I expect if we go around the room, it'll be the majority of people in this day and age. So normal muscle is this big component here from the white part, which is the tendon, through a muscle fiber. And if we kind of keep zooming in where that blue square is around one muscle fiber and zoom that in, it's surrounded by a muscle membrane called the sarcolemma. And again, if we zoom around where the blue area is, that's where dystrophin is localized. So exactly as Pat said, it serves primarily as a shock absorber, and it binds inside the muscle, and it binds to a set of proteins at the muscle membrane that keep the muscle's membrane's integrity intact over the course of lots and lots of contractions. And without it, the boy's muscles progressively get damaged. It's the pink, long, fibrous protein there right below the red sort of hemicircle that's indicated in this. I'll note also that the set of proteins that are up and above are called the dystrophin-associated glycoprotein complex. There's a quiz after this. And almost all of those proteins that are there, these balls and circles and squiggly lines, each one of those cause a different form of muscular dystrophy when mutated. So we know a lot about this. Over 40 different types of muscular dystrophies, well known and well characterized at this point. This is the dystrophin protein viewed in a bit of a different way, and this is how we think about it when we start looking at DNA mutations from individual children. So the left end is the start of the protein, called the N-terminal domain. The right end is the end, which is called the carboxy-terminal domain. At the beginning, there's a set of yellow areas, and those are the actin-binding domain, and that binds inside the muscle to what makes the contraction force occurs. And at the other end, the orange domain is what binds it to the outside part of the cell. And then all these blue parts in the middle are those springs, parts of the spring. And so depending on where mutations occur, it absolutely does have an impact on how Duchenne presents and manifests. You're going to keep me from waving my arms around? Very nice. Thank you. These springs that are in the middle, this is critical. You must be able to bind inside the muscle to the actin area, and you must be able to bind to the cell surface And these parts, it doesn't necessarily matter exactly how many springs you have. You could have 23, you could have 10, and they all can actually be largely functional. So I've got to introduce you to a core concept that you'll be hearing somewhat over and over again with different sort of analogies over the course of the meeting, is the central dogma. So this was worked out 50 years ago now. So this is a key component. Multiple Nobel Prizes were won in the decoding of this. And it's essentially, once we knew that DNA was the material that encoded what we inherit as double-stranded DNA, that goes through a process that's transcribed and goes from DNA into RNA. That RNA is heavily processed. So there's lots of parts of the DNA introns that we throw away. All the exons come together. And once it's in this RNA form, the machinery of the cell can read 3-letter codes. So every 3 letters of RNA equals 1 protein letter. So these 3 letters equal a P, these 3 letters equal an E, and then we spell out a word, which is a protein. So in this instance, it's peptide, which of course means protein. And so if we took one of these C's away at the beginning, we would not start at this CCT or this CCU, T's become U, and instead we'd start with a CTU, as we indicate down here, or CUG, and then read this, and that would translate into a completely different protein. So this is a frame shift, and a frame shift results in this protein that's called lusculum, and obviously very different than peptide. And this is a profound and common type of mutation that's observed in Duchenne. So most mutations for Duchenne result in a frame shift and lead to a completely dysfunctional protein. Dystrophin gene, the DMD gene, is incredibly large. It's one of the largest genes in the human body. It's encoded by 79 different exons, which are all crammed together here. And a common map that everybody looks at is a very nice representation where this reading frame is depicted per exon. So all of these exons, the 3-letter codes, the beginning of a 3-letter code and the complete end of a 3-letter code are the same within this exon. And in this one of the letters of this last 3-letter code is part of this next exon. So if the edges don't match up, then there'll be a frame shift. So a simple instance is if I removed exon 45, it would be a frame shift. If I removed exon 32, it would not be frame shifting. And these would have dramatic consequences, often for the function of the protein. So a more severe mutation, and this was established over the course of the last 25 years now, if exon 45 is missing, it's out of frame. And what that leads to is actually no functional protein being made after this point. And there's a nonsense mutation that's introduced here, which tells the protein to stop being made. And reading through that nonsense mutation won't help the disease because it would go to more gibberish, basically. So this is a truncating mutation, and oftentimes these proteins that are made actually get degraded by the cell or don't get transported to the cell surface at all. In contrast, there are some with big, huge mutations. This is missing exon 17 all the way through 48. Huge part of the gene is missing, and yet this person who had this mutation had a relatively mild disease, and that's because it's in frame. So this is the reading frame rule. And because this is in frame, even though it takes out a lot of this middle part, which are a lot of these springs, the disease is still relatively mild because it can be joined together. So what that looks like at the protein level is if you had that first mutation, just this tiny little bit, just take exon 45, so here's the numbered 1 through 79, take 45 out, it's catastrophic because it stops all the rest of the protein from being made at the protein level. If one happened to have this big, huge deletion of 17 to 48, though, much, much bigger than that. So at a genomic scale, it's a much bigger mutation. You actually end up with a protein that's missing this bit here, but this part is joined to that, and you actually have the key actin binding domain. You have a little bit of the springs, and you've got this key part that's at the cell surface. So this makes a largely functional protein as long as you make enough of it and as long as it gets to the cell surface. And oftentimes kids with Becker muscular dystrophy have these types of mutations. And of course, predominantly kids with Duchenne muscular dystrophy have these types of mutations. So when we look by Western blot, which is a mechanism to stain for protein, this is the normal amount of dystrophin that would be observed. And these are 2 boys with Becker muscular dystrophy where the dystrophin is smaller because a part of the middle is removed. And there's less of it as well because it's not as well made. It's not quite normal. And these boys have muscular dystrophy, but it's not as severe as this boy is likely to have, who has Duchenne and absolutely no dystrophin whatsoever at any size. And that's typical of almost all boys with Duchenne muscular dystrophy. If we look at the muscle a different way, so any of those of you who have had a muscle biopsy, we would look at histology, and this is not showing up particularly well, but we can see normal cells here, normal muscle cells. These are the individual muscle fibers. And around them, if we stain it with dystrophin, you'd see a bit of dystrophin around the edge of every single muscle fiber, really nicely staining here. In Duchenne, the muscles being damaged and broken down and degraded, and there's absolutely no dystrophin whatsoever. And in boys who are diagnosed clinically with Becker muscular dystrophy, often it looks like this, where there's less of this stain, but it's present, and the muscles are somewhat preserved. So most mutations end up being in this hotspot region of exons 45 to 55. Oh, goodness gracious. Sorry, my phone's beeping at me. They tend to be deletions or duplications most commonly. If they're not deletions and duplications, they're a category of nonsense mutations or frame-shifting mutations. So a single-base deletion, a single-base duplication, a one-base insertion, a one-base duplication would cause it to frame shift, the same as an exonic deletion often can, or it can be in the splice sites, so immediately outside of the exons, and cause the exon to be removed or altered rather greatly. So those are all categories. There's over 16,000 different mutations that have been reported for Duchenne, and so it's quite a spectrum. Because it's happening in every single generation and new mutations are coming every generation, almost every family, in fact, has a unique mutation that we don't typically observe in others. And when we describe it back at the exon level, so that people who have exon 45 deletions can all have different boundaries within the introns, for instance. There are some exceptions to this reading frame rule, and sometimes if there's deletions that are remaining in frame that are in this region that are important for binding to the outside, that causes Duchenne muscular dystrophy, even though it's in frame. And again, down here in the area that's for the actin binding domain, even though those mutations might be in frame, it can either be a severe Becker muscular dystrophy or Duchenne muscular dystrophy. And there's some other examples as well. Clearly, where the mutation occurs can make a difference. And this is an example of one paper indicating that mutations that are occurring after exon 63 are disrupting one of the 7 different types of proteins that are made from this one gene. And this one's called DP71. And that if DP71 is mutated, the boys are tending to have lower cognitive abilities. So shifts in diagnostic process, since this is all known, it used to be that history of physical determination of CK, a muscle biopsy was needed for diagnosis. Oftentimes, a muscle biopsy is never performed. I suspect many in the room have never had the child have a muscle biopsy. And since we can simply do a DNA analysis from blood, which is typically an array-based approach, or a PCR-based approach for the deletions and duplications, and if that's negative, then sequence the entire gene. There's still a fraction of the patients that even when all of that is done, we still don't know the causal mutations. And there's some new molecular tools, including whole genome analysis, that will solve many of these cases. So this is an example of solving a case where we could not figure out the mutation by every known clinical test. And in this instance, we sequenced RNA from a small amount of the DNA, a small amount of muscle that was available from this child. And sequencing indicated that there was no expression after exon 37, and that this was spliced to a region that was 5 megabases, 5 million bases away. At the DNA level, we could see that there was an inversion by doing whole genome sequencing. We could search the entire genome for what was going on. It turned out that this 5 megabase region was inverted, which is a very rare type of mutation to have in Duchenne, but has been reported. And this breaks the gene here in intron 38 and leads to a mutation that's not possible to skip. All of the parts of the protein are there. They're just in the wrong order. But what's important here is that by finding exactly where this breakpoint exists in this particular family, we could design primers on the left and on the right and design a simple PCR test that's now a family-specific method for doing the diagnosis in this family and could be used for the women in the family for carrier testing. And that's an important aspect to be able to follow up on in this. So obviously, we should all care about mutations. it does greatly impact and improve our ability to understand the disease, and actually studying some of the kids that are unusual is going to be profoundly important in this coming decade. It also leads to much more specific information that for genetic counseling, and is now obviously quite important for being sent to appropriate clinical trials. Female carriers for Duchenne muscular dystrophy are often not manifesting and lead completely normal lives. There's an increased risk for cardiomyopathies, and a fraction of females do manifest the disease, and some manifest the disease equally as severe as Duchenne muscular dystrophy. So it's actually quite important to know this, and I would argue to know it relatively early in females. The best disease frequency is it's about 1 in 5,000 in males in the population. The mutation rate is very high. It's the most commonly mutated gene, and that's why we're seeing it over and over again, and that's why we see it often completely new in families. The mutation rate of 1 in 10,000 is at least tenfold higher than most other genes, and that's why we certainly observe this common scenario where mom doesn't know that she was a carrier, and she doesn't know that her mom was a carrier, or alternatively, her mom may have been a germline mosaic and had a fraction of her eggs mutated. And oftentimes we observe a de novo mutation in the child. The mother's blood doesn't have it. The father's blood would not have it because the X chromosome came from the mother. And the mom is then determined to be a germline mosaic and have a higher risk than the general population of subsequent pregnancies. So we've used data from Duchenne Connect, which I hope you've all registered for. Holly PA is going to come on after me, and I hope you go to the Duchenne Connect booth. It's a spectacular resource and has the promise to be even better into the future. From this data set, I wanted to just show you one part of this, was that we certainly can look at the age at which boys are ending up in wheelchairs. So some boys are needing a wheelchair at age 5, 6. Others, with no dystrophin whatsoever on their muscle biopsy, are still walking at 17, 18, 19, 20. So very different presentations of the same disease, both missing dystrophin completely. So why is that? It could be these people got spectacular therapy, and these people got dreadful therapy or did things that were bad, right? Unlikely. And as a matter of fact, when we look through all the different things that they've done, we do find that there's a set of things people have done. Obviously, steroids help prolong the age at loss of ambulation. And when we look at different treatments that people are taking as well, vitamin D, coenzyme Q, those are predictive of longer age at loss of ambulation. And there's others that are suggestive, but they don't account for these dramatic differences in severity with more severe and less severe kids. So the idea is to look at these kids and search very intensely for, are there any genetic mutations that might be causal? So exome sequencing or whole genome sequencing is already in clinical practice, where we search throughout the genome to try to find the one base mutation causing a spectrum of over 5,000 different rare Mendelian genetic diseases like Duchenne. We can use those same tools to try to ask, does a given boy with Duchenne have another gene that's mutated that's making it more severe, or another gene that's mutated that's making it more mild? We learn about these proteins then as possible modifiers of the disease process and may lead us to new therapeutic strategies. This is possible because in the last 10 years, since the first human genome was sequenced for a cost of $3 billion, the cost of sequencing has dropped by 100,000-fold. So this actually brings it into a realm where we can afford to do essentially whole genome sequencing for about $2,000 to $3,000 per person. So there's a genetic modifier study. People are very welcome to participate in that, and I can share information with that. We're specifically trying to sequence the people that are at the extremes of the phenotype for that study and identify genetic modifiers. There's a couple that are described in the literature, for instance, osteopontin variants by Pagararo et al. And Melissa Spencer has shown some therapeutic relevance for this finding. And LTBP4 variants that Kevin Flanagan and Beth McNally had identified in humans as a modifier, and Beth McNally is working on it as a therapeutic lead as well. So can we find a richer set of such types of therapeutic leads from this sort of broad-based search? Data looks like this, that we find about 200 protein-damaging, completely new mutations in every single person's genome that we sequence. And it would be true if I sequence every single person in this room. All 200 of them are unlikely to be playing a profound role in Duchenne or in your life. And so what we search for are variants that, in this instance, a variant that might be present in a more severe brother and absent in a less severe brother. So in this particular instance, there was a difference in this gene AMPD1. Defects in AMPD1 cause AMP deaminase deficiency, which is a metabolic disorder that affects muscle function. So it may make sense that this variant could play a role in this particular family. We search for plausibility that a specific rare variant, possibly new and possibly only in that one family, is likely, and then we need to develop cell line models to figure out if indeed there's a causal role for that specific mutation. So I'll give you a little taste of what these data look like. We actually can look to see that in these data from the first small wave of boys, there's 6 independent very rare mutations in a gene called LTBP4, which is the variant, which is the gene that Beth McNally had identified. And these may indeed give us some insights into how does LTBP4 variants and the broader spectrum of variants play a role in modulating the disease process. And we'll learn more about those in the coming year. So I just want to end by summarizing that everybody needs a diagnosis, a molecular diagnosis. It can account for a fraction of the information, and we need to be alert for finding those people with unusual disease courses to learn more from them. And I think we've got a great resource by registering at Duchenne Connect to allow scientists to connect with those individual families. And I'll stop there with just showing the acknowledgement slides. Appreciate it.